right. Can you guys get out your notes? I want you to take notes on this part because this is like, I'm going to answer the questions that are left. Okay? How was that? Were you guys okay? Did you feel confident in your lab skills or not?
Yeah.
I was pretty much like satisfied with the way you did it. No major problems. But I do think that there's not enough communication and like group work, right? Like you guys are kind of all like just, like moving around each other, and then like at the moment you're just like, oh, I'll take the cash off, or oh, I'll do this, or oh, I'll label it, right? There needs to be a little more of like planning and like more, like systematic execution of these steps, right? Because otherwise, like you and all these other groups are getting mixed up. You need to figure, kind of divide roles, right? And that's not something that I should have to do. You guys should do that among yourselves, right? So you can choose a group leader from now on and be like, all right, you can be the group leader and you can tell everyone else what to do. Or you can say that someone's always going to be the recorder, someone's always going to be like the transfer of things. Like, I don't know, just think about that in the future, because I do like to see those types of skills in labs, right? Okay. So we're going to talk a little bit about this thing today. Like basically, what's the point of this? Why do we do this? I need to find the PowerPoint though. So. And I'm sorry for not posting the PowerPoint yesterday until late. I'll try to. This one I can post right away, but you should have it on your diagram too. Okay. And anything that we do in lab, you guys know, is like fair game on a quiz, right?
Yeah, please.
So if I were to ask you, what Benedict's solution tests for, what would you tell me, Mike?
Glucose.
Not just glucose, but it tests for any monosaccharides, basically, right? So glucose, galactose, fructose, right? And then if I asked you what color a positive Benedict's test would be, what would you say? You would say brick red, right? And then if I asked you, any other details about, the lab, you would know, right? Okay, good. All right, so let me see. So the title of today's, this is really brief, and we're just going to fill out that little chart at the end of your, packets, right? So this thing is called glucose homeostasis. I didn't write it on here, but maybe I will. All right. Oh. Okay. So first of all, what does homeostasis mean? Do you guys remember? Arsh, I think, had his hand up first. I'm sorry.
It's like the regulation, like the reactions that regulate like certain systems in the body, or like reactions.
Okay. We need to be a little more specific.
Maintaining a stable internal environment even when the outside environment changes.
Perfect. So what were the different characteristics that we needed to maintain consistent? Things like what? Temperature, right? We don't want to get too hot or cold. Even if it's freezing outside, we don't want to get freezing also. What else do we need to maintain internally? The amount of salt, the amount of pH, so the amount of pH level, or the amount of sugar. And that's why we're talking about this right now, right? Do you want too much sugar in your body? No, because what can happen? Well, diabetes is a condition where you have too much sugar in your body. It can just ruin your organs. It's just having too much sugar all over everywhere is not a good thing, right? So we need to manage our level of glucose, right? So here's this diagram that kind of shows what happens in our body when we eat, right? So we're connecting this lab to the process of eating, digesting, and then urinating. Okay? Fun. All right, so first, what do? You're eating something. What do we eat a lot? We eat starch, right? So we're eating starch, chomp, 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 and then starch starts to get digested in our saliva, right? And then, but after it starts to break down our saliva, and then it goes into our stomach, and we start digesting it, right? And what does starch break down into? Does anyone know?
No.
Can you guess?
Carbohydrates.
Starch is a carbohydrate. It's a huge carbohydrate, and it breaks down into monosaccharides, right? And what's the main monosaccharide that we love to talk about? Glucose. So we have starch. We break it down into through digestion, right, in our stomach, into glucose, right? And then all those little glucose monosaccharides, right, are traveling around in our intestine, and they get absorbed into the bloodstream, right? So now we have glucose floating around in our blood. Is this a good thing or a bad thing? Do we need glucose to live?
Yes.
Yeah, so we need glucose in our blood. That's good that we can get it into our blood. But then who really needs it? Do we want it to just like run around in our blood all day? Where do we need it? Who's using it? The organs, but more specifically, what are organs made of? Cells. The cells need glucose to do what? To survive. But why do they need? They need it to function, but what does it make for them? Energy. They need energy to do anything, like any sort of task that a cell does. It needs energy. Where does it get the energy? By breaking down glucose, right? Okay, so we have glucose in our blood. How are we going to get it into our cells? It can't. It's too big. It can't fit across the cell membrane. So what do we need?
Those things that
We need a magic little sort of tunnel that's going to transport it across into our cells, right? But how does that tunnel get into those cells?
I forgot what it was called. It's something. The vacuoles that absorb.
Got it.
That's a little different. The vacuoles actually don't make a glucose vacuole. It's actually glucose itself is transported by what?
Endocytosis.
No, that would make a vacuole too. So no. They're actually transported by so if this is our cell membrane here, right? Basically what happens is these transporters, usually it doesn't let glucose across. Let's say here's glucose, right? We know that it looks kind of something like that, right? From yesterday's model building. So it usually can't go across, but then in order to get it across, we have to make this transporter, which will kind of let it go across. Do you know what makes that transporter available? And you guys have all heard of this. It's a hormone. So insulin is a hormone. I want you guys to know that it's a hormone, right? And it's made in the pancreas. But it's made by these specific cells called the beta cells. And beta is like that sign right there. It kind of looks like a weird...
It's like a bee, but a bee.
It's like a bee without this tail on the top, and it kind of starts from the top and kind of goes like that.
Like a bee and a bee had a baby.
Like a what?
Like an uppercase P.
And a bee.
And a bee had a baby.
Are you saying it looks kind of like a pregnant person?
No, like a bee in the letter B and the letter P had a baby. So it has the stem from the T, and it has the two bumps.
Okay. But it also doesn't have this tail up here. That's what I'm talking about.
Sick.
But it does look. I mean, do you see my confusion? Baby starts with B, and then this looks kind of like a. I don't know.
Oh, whoa!
Is that where you're? That's what I thought. I thought he was being really deep, and I was like. But no, okay. Kind of. All right. Anyway, so if we once insulin gets in the picture, so here, let me just draw this again. So here's our bloodstream, right? And then here, let's say here are some cells, right? Here's the nuclei right there, right? So then so here's glucose, right? Originally it was in here, right? And then it usually can't go in here, but when insulin is there, insulin does what? It makes these tunnels, right? And then the glucose can go in, right? So what happens to the levels of glucose in the blood when insulin is there? It decreases, right? And because it's now all in the cells, right? All right, we're good so far? So now we have the glucose in the cells. That's good. And what will the cells do with it? There are two possibilities. They'll use it, right? Why do they need it? When do they use it?
When they carry it.
What process uses glucose?
Cellular respiration.
Go.
Let's go.
So one way is it provides energy.
Why do we use it?
All right, so we have. So the cells could use it immediately if they need it right then. They could be like, all right, mitochondria is going to break it down into ATP, right? And then it can be used in cellular respiration. What if we ate a lot of starch and we have a lot of glucose there? Yeah, well, we can't use it all right then, so we're going to store it. Does anyone know what form we store it as?
As lipids.
Fatty acids.
We store it in a polysaccharide form, and it's called glycogen. Have you heard of that? Yeah? So glycogen, and that happens in the liver, right? The liver basically takes all these liver cells, right? They take up all these little glucoses, and they make this like branched model, this branched thing like this, right? And that's called glycogen. And this is just the way they store it. That's like their storage like method, right? So this is glycogen. Glycogen looks like that. And it's really all of this is all these little glucose monomers,
So that's simple.
right? And they're all coming, Anyway, you guys get the picture, right? All these glucoses they're putting together to store it for later, right? Okay. So then suppose like a few hours pass, right? We're kind of starting to get what now? We're starting to get hungry again, right? Or we're starting to like lose all this energy that maybe we used up all the glucose that we had already. So what do we need to do now? We need to either eat again, or we could take this stuff and use it. So what do we have to do? So instead of storing the glycogen, now we have to do glycogen breakdown, right? Do you think insulin does that too? No, insulin gets the glucose where?
From the bloodstream.
From the bloodstream into the cells. But now where do we want the glucose to go? We want it to go back
Into the bloodstream.
into the bloodstream. So what's going to get it back into the bloodstream? It's another hormone that's also made in the pancreas, but it's called glucagon. Do not confuse glycogen and glucagon, right? They're two totally different things. This glucagon is what? It's a hormone, right? Made in the pancreas.
Or what kind of society?
Made in the pancreas by what kind of cells?
No, not about.
The beta cells make insulin.
Omega cells.
So, who said what?
Alpha.
Alpha cells, right?
Or,
Right, so now instead of storing the glycogen, we're actually breaking it down. So now we get the glucose monomers again, right? Sorry, I'm just being lazy and I don't want to draw all 6 sides of it, but it's and here's the O here. But anyway, so now we have all the glucose, the individual glucoses here, right? And then the glucose will be back in the blood and can travel to other cells that might need it, right? So which hormone is responsible for lowering the blood levels of glucose? Mike?
The
Insulin, right? And then which hormone is responsible for raising the blood levels of glucose? Arsh. Glucagon, right? What's glycogen?
Stored glucose.
So glucose is how many molecules is that? One, so it's called a what? It's a monosaccharide, right?
It's a polysaccharide.
So glycogen is tons of, thank you, and this is a preview for the next few days, right? So glycogen is basically a ton of these all branched out like this, right? So this glycogen is this is a polysaccharide.
I was asking how to call
Many saccharide sugar. Good? So you really have to know these different, where they're made, right? Alphas, this one's made of alpha cells, both in the pancreas, both hormones. This is a polysaccharide, this is a hormone. Don't confuse those two words. I'm telling you, it is confusing. And if you write something that's like glucogen, that's like a hybrid of both, I can't accept either of that, so I need you to make sure that it's glycogen, glucagon. Charlie?
Where it says stores glycogen, e.g. in liver, does that mean it's being stored in the liver?
In the liver,
or
in the
it's
liver
being
cells.
is it being made in the liver cells? Like, is it being turned into glycogen?
It's stored in the liver as glycogen, so it's in the liver. Yeah, that's happening in the liver. All right, are we good so far here? No? Yeah. Oh, I thought someone said no. Okay. All right. So now, okay. So this cycle, you see how these like maintain the homeostasis. So if the blood sugar levels get too high, insulin's like all there, ready to get the glucose in the cells, right? If it gets too low, if it drops too low, then we need to like access these stores, and then glucagon will bring it back up. So they're like constantly detecting what the blood sugar levels are, and then it's maintaining homeostasis, so you live in this like really narrow range of like acceptable values for your sugar levels, right? Good? Okay, so you see this cycle. All right, so once we have glucose in the blood, we can also, so we take it in the blood, and then what happens to the excess of it? It can't all necessarily be absorbed, right? So what's going to happen? Some of it sort of gets through and goes to where? Where do things go after we've... The kidneys, right? It goes to the kidneys. So now we have glucose in the kidneys, right? And the kidneys will also try to reabsorb some of it if they can. Reabsorb it back into the bloodstream so then it can keep going on around that cycle. But what happens if there's too much for the kidneys to even reabsorb? Then where's it going to end up, Mike?
The urinary system.
In your urine, right? Is that good or bad? It's not good to be like losing all this glucose because your body should be absorbing it and taking it in, right? If you have diabetes, that's a deficit in, does anyone know? Insulin production. So insulin production in your pancreas by your beta cells, somehow it's not working, right? Whether you're born with it, right? Some people have type 1 diabetes, right? Which means that they were born with it. Type 2 means that they've sort of acquired it over the years. And, I think that a lot of it is lifestyle factors. So if you're constantly overloading your system with sugar, right, then what's going to happen to your pancreas? You're going to keep on making all this insulin, and what's going to happen?
It's like not going to be as efficient, and it's going to slow down.
Yeah, it's not going to be as efficient, right? So you're trying to make all this insulin, but it's like not really, it can't keep up with all the glucose, right? So if glucose, let's say that insulin somehow, if there's a problem with this system right there, then where's the glucose going to stay? In the bloodstream, right? And then we're going to have more glucose here, right? Because it's in the blood. And then we're going to have more glucose in our urine, right? Do normal people have glucose in their urine?
Yeah.
Not a lot, right? You shouldn't have glucose in your urine. What's a way we could test our urine to see if we have glucose in it? Add some Benedict solution, like, heat, what did you, constant agitation?
Yes, constant mild agitation.
Mild agitation. So if you're diabetic and you go and you give a sample of urine and then they put Benedict's in it, what would you expect? What color would you expect it to be?
Red.
Brick red, because you would have monosaccharides in your urine, right? So that's a good way to test. That's a way that they test whether someone's diabetic or not, right? Interesting story.
Cool.
Any questions?
So, like, when you're on a sugar high, you have, like, a lot of energy, but then, like, when you crash, it turns out.
That's a good question. I mean, like, when you're on a sugar high, your cells are all, like, using all this.
There's no more.
This, so I guess they'd be, like, over, like, stimulated, but then if this is working to also, like, deal with that excess glucose, right, it would be storing it, and then once it's out of the bloodstream, you're kind of just like, wait a minute, I need that back, right? So it kind of like, it's like a delicate balance, but I don't know if it's like that scientific of like a process, but yeah, basically you get this surge of energy because all your cells have the glucose, and they're like, okay, we can do whatever we want, and then after it's gone and stored or just used up, then you're like, oops, yeah. Mike.
Is excess glucose stored in urine form? Is like excess glucose stored in urine?
It's not stored. Urine is something else. It's like
form.
Excretion.
Huh?
Like the urine form. Or form, just like the form. Like what form is it?
It's just glucose, but it's in your urine. So it's like if you had glucose, like that powder in your urine. It's like your urine is going to be your urine no matter what. And it's not like you're changing glucose into urine, but it's just more glucose is coming out into your urine because you're not filtering enough in your kidneys. Right? All right, we're good?
Yep.
All right. I want you guys to kind of just sit like stay here and not crowd around the door before we leave. All right? Does that sound like a good plan? Who needs? I will. Oh yeah, I'll like pass out your phone. Whose is this?
Oh, Rose.
Huh?
Rose.
You got one?
Whose is these?
Two. I want to put my money on this one's Jasmine, so that was the newer one.
No, it's under. Oh, it's under the.
This one?
No. Yeah.
Amazing. Wait, yours is this one?
Who's this? Anyone?
Yeah, no, the other one's Jasmine.
That one's fine.
Yeah, this one's Jasmine because I know Jasmine got this one.
She walks up to his kid, he's like, you are going to eat something and George. And he just goes like, tomato, like,
Otter, Fox,
and everyone in the class is
Under Armour.
like, no, I'm just kidding, your mom was and it's so funny. He was so scared. Yeah,
All right, who didn't get theirs back?
Nothing done. I'm on my visa system.
you're holding mine in your left hand.
Imagining and math.
Okay. You guys should be studying every night. Whose is this?